My name is Charlene Velichko, and I'm a Scientific Director in the Eurofins Discovery Phenotypic Services Group. I'll be presenting a short case study on the use of the BioMAP Phenotypic Platform to pivot and reposition existing assets to address a new disease indication, such as a fibrotic disease like idiopathic pulmonary fibrosis, or IPF. While repositioning is an excellent opportunity to leverage existing assets with favorable PK/PD and safety profiles for new indications, as well as potentially extend the patent life of these assets, repositioning is not without its own set of challenges. One of these is indication selection. Combining phenotypic and target-based information can help to streamline hypothesis testing, expand to potential new therapeutic areas, and increase the chances of clinical success for repurposing programs. When considering a small library of drugs eligible for repositioning, systematic testing in a well-validated system can help rank candidates for a given indication. Likewise, when considering potential partners for drug combination approaches, the same systematic approach can be applied. In this case study, we will demonstrate how a small library of repurposed compounds can be quickly evaluated and prioritized for a new indication, namely IPF. Evaluating compounds in a platform that maintains a complex etiology of fibrotic disease while using a robust, scalable format can help ensure lower attrition as test agents move along the development pipeline. Additionally, as this library represents different target classes and MOAs, the ability to compare compounds head-to-head in a physiologically relevant but target-agnostic manner is critical. The approach taken was to perform broad phenotypic characterization of the two approved IPF therapeutics, pirfenidone and nintedanib, using the BioMAP Diversity Plus panel. Although these agents represent very different MOAs, they do share common phenotypic effects, which we can consider sentinel activities. As these may play a role in their shared efficacy in IPF, we sought to identify other compounds within the library that share these sentinel activities and to rank the compounds based on the overall number of shared activities. The top-ranked candidates were then further evaluated in a more fibrosis-focused panel. Based on this approach, one is able to quickly rank and validate compounds for antifibrotic efficacy using a rationale based on data from physiologically relevant human primary cell-based assays. This represents an efficient and low-risk approach for companies interested in rationally expanding their drug pipelines for new indications with an unmet medical need. This slide shows a comparison of the Diversity Plus profile of pirfenidone versus nintedanib. The Diversity Plus panel contains 12 different assays, which we call BioMAP systems, that are denoted by the circular icons, and a total of 148 protein-based biomarker readouts. Using specific criteria, we identified 25 shared activities out of the 148 total analyzed biomarkers. These shared activities, which we consider to be sentinel activities, included anti-inflammatory activities, anti-proliferative effects, as well as antifibrotic effects. We then compared the relative activities of the library compounds on these 25 biomarkers and ranked the library compounds according to the number of shared activities. Using this approach, Agent L was prioritized for further evaluation for fibrosis. In order to further elucidate the effect of Agent L in tissue-specific fibrotic assays, we profiled this agent in the BioMAP Fibrosis panel, which consists of 3 systems. All 3 systems contained myofibroblasts, a cell type that is central to the pathogenesis of various fibrotic diseases. The myofibroblasts are either in monoculture, seen in the central panel, or in co-culture with epithelial cells from either the small airway, as seen in the panel on the left, to model pulmonary fibrosis, or proximal renal tubules, as seen in the panel to the right, to model renal fibrosis. Profiling across these 3 systems confirms that Agent L is able to decrease alpha-smooth muscle actin, a biomarker associated with myofibroblast activation in 2 of the 3 BioMAP systems, as well as decreased collagen production and other biomarkers associated with fibrosis. This is consistent with an antifibrotic profile. However, in comparing the impact of the 2 co-culture systems, agent L was cytotoxic in the system modeling renal fibrosis at higher concentrations. Renal cell cytotoxicity can be a red flag for safety, so if these concentrations are needed to elicit antifibrotic effects, this is an early indication that systemic routes of administration may be problematic at this dose range. We have 2 types of services that are based on our human primary cell-based assays. BioMAP profiling services are geared toward projects with smaller numbers of test agents. The deliverables for profiling services include a PDF report, along with comprehensive analysis for insights into MOA and biomarkers related to efficacy, safety, and indication selection. For screening and prioritization of larger numbers of compounds, we have our early screening services. The deliverables for screening services are streamlined and include data tables and heat maps needed for higher throughput evaluation and quick triaging of problematic compounds and prioritization of compounds based on expected biology. The fibrosis early screening, or ES services, start with a minimum of 72 compounds, which will be screened in 1 or more of our fibrosis systems, depending on your program needs. These compounds are screened at 4 concentrations in singlet. Compounds that were cytotoxic or inactive in the selected system can be immediately deprioritized. For the remaining compounds, these can be further ranked based on the total number of activities, as well as the total number of activities associated with antifibrotic effects, as well as anti-inflammatory effects. In summary, Eurofins Discovery has multiple options that can be exercised alternatively and tailored accordingly based on the needs of the client's program. Clients may choose to initially screen a library of compounds in our broader Diversity Plus panel in order to rank compounds for their potential in multiple indications, as well as flag compounds for signatures related to potential clinical adverse effects. If some of these compounds have initial profiles that look promising for fibrosis, these can be further prioritized using our fibrosis early screening services. A more in-depth Diversity Plus profiling project can be done with select exemplars or chemistry lead compounds to further elucidate MOA. Thank you for your attention. If you'd like to contact me, my email is charlenevelichko@eurofinsus.com. You can also reach out to your local Eurofins Discovery representative or visit our website at www.eurofinsdiscoveryservices.com and click on BioMAP. Thank you for your attention.